Great, welcome to my video project. I'm going to start by showing how to send out samples to an outside laboratory. So I have blood that we drew from a dog here, Carmel, and I'm sending out adult wellness, which for us is a CBC and a chemistry 10 profile. So I already spun down the chemistry. It was in a red top tube and now it's serum in the tube. Place the correct label on with the correct patient, doctor, and what test I want. And then I put a little parafilm on top just to make sure there's no leakage. CBC sample goes in the purple top tube to prevent clotting. Same thing, I've got it labeled, and then the parafilm. We do all of our forms online when we send out blood work, so I went online, entered the correct doctor, the correct patient, the test that I'm looking for. So then what I do is I fold this up, put it in the bag along with the samples, and I keep a copy here too so that when it comes back we know what exactly we're looking for. We write it down in a book to make sure that we have log of everything that's going out to outside labs. So I stick those in here all together. I always double check again to make sure my patient on the paperwork and on the labels all matches before I send it out. And then this would be refrigerated. So biopsy samples are very similar. These are the samples from the necropsy that you'll see later in my video project. Since they're in formalin and that can be caustic for people, we're just going to put a little bit of tape around that to prevent spilling. Do that here for both samples. This is a liver and a spleen biopsy. And then for this paperwork, the doctor fills it out of what they're looking for. So I have a sheet here that just goes through all of the patient's information, what kind of samples we're sending. We're sending a histopathology biopsy. Doctor writes a quick history of what's going on. And then I'm going to tear off the bottom copy. And we'll save that for us here. So same thing, when the results come back, we have a copy. I'm going to fold this and put it in my sample bag here. And then the actual biopsy samples I'm going to put in a separate bag away from the paperwork. So those are going to go in here together. I'm going to seal this bag up. And then I'm going to take this bag and double bag it within the one that has the paperwork. And this again is just to prevent spilling and contamination with the formalin. Okay, and then once that's in there, zip this bag up too. And then we have a refrigerator that's reserved just for lab samples and lab equipment. So that is around the corner here. And then I'm going to place these in the refrigerator, and then the NTech will come pick up both of those. That's it. So we have two main machines that we use in hospital for our blood work. We have our CBC machine and then our dry chem chemistry machine. So I'm just going to quickly talk about the maintenance for the chemistry machine. So first thing we do is once a month we run a control. And what that is, these are refrigerated. We take 5 mils of sterile water, place it in here, gently mix it. This sets undisturbed for an hour. In the interest of time for the video, obviously I'm not going to do that now. But once this has been sitting, mix it gently and then put about a half a mil into this part here. We have two different sizes. There's a larger one, but we're going to use the smaller one for this since it's a smaller amount. This is where my slides go. So for our control, we usually test electrolytes and the pre-surgical chemistry. So those are set up in there. And I make sure I have my tips in for the electrolytes and the control. And then I would close the machine here. I would set it up here. It has references for dog, cat, horse, other, and I would change it to control. And then I would press start, and this would run through everything. It would print me out a list here of the control, and then I would import results on the computer. We do keep the manual here, so if there's ever any issues or anything like that, we can try to troubleshoot it. As far as cleaning the machine goes, we have in here, these are where the samples from the day and the tips come out, so I would empty those into the garbage. And then if this is looking a little bit dirty, I can clean it with a Kim wipe. The filter I would take out and clean that with a Kim wipe as well. So I would just take this out, kind of just gently wipe it, and then place that back in. The other parts of cleaning that are important for the machine that are only done once every 3 months is cleaning the mirror and the incubator, which are located in here. I'm not going to show that because we only do it when necessary. If we overdo it, we can actually do more harm to the machine than good by doing that. So once my results are imported for the control, I'm going to put them, we have a logbook here where we log each one, make sure the reference values are still correct, make sure everything's functioning well, and we keep a record of that. So I'm running a parvo test on a puppy that came in for vomiting and diarrhea. We want to make sure it's parvo negative. So we took the sample from inside the anus, just a light swab. We don't want to get too much fecal material on it. Then I place this back over, invert the tube here, and then break the purple part there. Then I'm going to squeeze this 3 times, and then remove the swab. And I'm going to place 5 drops of this onto the well here. And then once color appears in the small dot here in this well, then I will snap it down. And there it is, I can start to see it. Now I'm going to set a timer and wait for 8 minutes, and then we'll read the results. So it's been 8 minutes, our timer just went off. So we can read the results now of the test. If we compare it to the chart that comes with the kit, it is a negative result, which is great news. We can tell the owner that their puppy does not have parvo disease. So this is Brutus. He's here. We're going to do an ear smear and a skin scrape on him to check and see if he's got anything going on. So first I'm going to do the ear smear so we can check to see if he has any kind of infection. We're looking for yeast or bacteria. So what I do is I take my swab and I'm going to put it down into the ear canal and just kind of gently swirl it around a couple times. And then since that's his right side, I'm going to break it so that I remember which side is which. And do the same thing on the left side. Good boy, Brutus. All right, and then what I'm going to do is I'm going to take both swabs and I'm going to put them on the same side. Always put the left towards the frosted, so remember which is which. So I'm just going to kind of roll it back a few times. And then do the same thing with the right. And then we'll move into the lab and we'll stain it. So I'm going to stain the slide for Brutus's ear smear. So first I'm going to heat fix it, light a match here. And then I'm just going to swipe this underneath the flame just a few times. I don't want to actually char the slide, I just want to kind of get rid of the earwax that might show up on the sample. And then I've got my stains here. So what I do is I put it in each one for about a minute, let it sit. So while that's sitting, I'll talk about what I might find on the slide. I might find yeast or bacteria, and we categorize them based on a 1+, which is slight, 2+, which is moderate, and 3+, which is severe. So I'll look through a few different fields and kind of get an average of what I'm looking for as far as yeast and bacteria. If I were doing a mite check, I would actually just put some of the earwax right on the slide, put some mineral oil on, and just look at it without staining it, but we're not suspecting ear mites, so I'm not going to do that. So I think it's been about a minute. Next one. I'll just rinse it gently and then dry it with some babyless paper. Just keep looking at the clock to make sure I'm keeping time. So I'm going to go ahead and just rinse this. Dry it off. Okay, and then I'm going to head over to the microscope here. And what I'll do first is I'll focus it under willpower. I'm going to place a drop of emergent oil on the slide. And then look at it under high power in the oil. Each ear to see if I find. So far it looks mostly just like debris. Brutus has been itching at his ears some, but he is an allergy dog. There's a little bit of cocci, maybe 1+. That's the left side. All I'm seeing, so then I'll go over to the right side. Same thing there, just about 1+ cocci. So I didn't see any yeast, don't see any rods. So once I give the doctor the results, they'll decide how to treat it. So this is the necropsy section of the video. As you can see, I'm wearing my personal protective equipment. I have on a gown. I would either wear a gown or a lab coat or some kind of scrub top covering my normal clothes. I also have a face mask on and a set of protective goggles and a set of gloves. If this cat were a rabies suspect, I would wear 2 sets of gloves, so I'd be double gloved. And it would be really important when handling rabies suspects that both the doctor and the technician involved are vaccinated, which I am. And then these are the tools that we would use for the decapitation portion for rabies suspects. We'd have a more protective set of eyewear here, the double set of gloves, and then the different tools. What we would do for the decapitation would be to bag the head in a black bag and then double bag it inside of one of the red biohazard waste bags. The head would then go in the refrigerator. We would call the health department, and the health department would come pick up the sample. We would hold the body in the freezer until the health department gave us the okay, then we would dispose of it as per the owner's request. So fortunately, Alex here is not a rabies suspect, so it's just a regular necropsy. We're going to collect some samples to send out. These are the tools that we typically use. We have the scalpel, which is what we're going to use most often, and then the different forceps and the hemostats to kind of hold things out of the way as needed. So we're going to go ahead and start. Dr. Lace is going to make a ventral midline incision. And then we'll come back to the video. While she's doing the necropsy, I'm going to fill out a form here, just going through all of the different parts of the body, writing down what we find. If anything is abnormal, we'll write that down, and if there's no findings, we want to make sure we write that down. We don't want to leave anything blank. So we'll be doing that throughout, and then we'll come back to the video once we're collecting samples. So Dr. Lace took samples from Alex, our cat here, so I'm just going to prepare them to send out to a lab. So what I'm going to do is take about a 1 centimeter slice here from the liver. And I want to make sure I have 10:1 formalin to sample. So then I'm going to put this, stuck to the gauze, in the jar there. It's already labeled that this is Alex and this is his liver sample. And then same thing with the spleen. That in the jar. I'm going to take the gloves off to the top on there. And then with these, we send these out to an outside lab. So I have the form here from Antech that I'll fill out. I'll fill out all of the findings that we found in the biopsy, mark where we're taking it, fill out the client's information, and then we'll send those out and they'll come pick them up. So this is Maggie. She's anesthetized. She's intact. She's 9 years old, but we're going to spay her today. So before we do that, we're going to take vaginal cytology. So Dr. Sorg is just thoroughly cleaning the area before she takes the swab with Chlorhex and saline. She's cleaning a few times to make sure there's no contamination. Next, she's going to take a sterile cotton swab and then moisten it with some 0.9% saline. And that's to prevent cell damage. So she's going to insert the swab almost completely vertically. And when she's in past the sphincter, she'll turn it horizontally and gently roll it around to get the cells. And she's going to hand it to me, and I'm going to make the slide. So I'm going to roll this gently 3 times in 3 lines. And then that'll take a little bit to dry, and then we'll stain it. So I'm going to stain the slide for Maggie cytology. We took a vaginal smear. About 15 dips in each one. I'm just going to gently rinse with some water. Dry it over here with the Vitless paper. And we'll head over to the microscope and take a look. So first I'm going to focus it on the dry, and then put a drop of oil on and switch it to the immersion oil setting. Definitely see a lot of the parabasal cells. Mostly parabasal and intermediate cells. I'm going to make sure I read several fields from each line. I'm not really seeing any red blood cells or debris. Based on the cells in here, I would say she is in the anestrus stage of the estrus cycle, which means that there's no behavioral changes, she's not ready to reproduce. And that would explain seeing the parabasal and the intermediate cells. If she were in proestrus, I would see intermediate parabasal and some superficial cells. And that would be an increase in the estrogen, but she would not be, males would be attracted to her, but she would not be ready to present for coitus. And if she were in the estrus stage, that's when we would start to see mostly just the superficial cells that are keratinized or cornified. And at that point, she would be ready to go ahead and present herself to a male for mating, and the estrogen would decrease, the progesterone would increase at that point. After that stage would be the diestrus stage, where the intermediate and parabasal cells would come back. We wouldn't really see a whole lot else, just a lot of debris, which is why the anestrus and the diestrus phase can be hard to differentiate, but in the diestrus there's usually more debris. So based on the slide, we'd say Maggie's in anestrus. So this is the DNA cross-matching part of the video. So I've already drawn blood from 2 dogs, Carmel and Brutus, and I collected the samples in purple top tubes and then the red top tubes, spun the red top tubes down to obtain the serum, and the serum is now in these red tops. And I already just spun down the purple tops, and I'm going to decant the plasma off of both of them. So this is Carmel. She's going to be the donor, so that's the major or the minor cross match. And what I'm going to do is just take a few drops of the packed red blood cells, and I'm going to add a few into here, about 3 drops. One more. And then I'm going to fill the rest of that tube with 5 to 10 mils of saline.
Thank you.
Try to do this all without making a mess. Okay, I'm going to do the same thing with Brutus's sample. He is the recipient. So I'm going to decant the plasma for his. Remove that. And then same thing, just a few drops of the packed cells. And then I'm going to add saline to that as well. Making sure they're about even. So then what I do is just gently kind of mix them, just by kind of swirling them around here, and then I'm going to centrifuge them for 1 minute on about 350 or 3,500. So place both tubes in, make sure they're even so that they're balanced, and then turn this on just about 1 minute, and then we'll come back. So I'm done centrifuging the packed RBC cells, so what I'm going to do for both is just pour off the supernatant. And then I'm going to go and resuspend both with more saline. And then I'll spin it down and repeat this 2 more times until the packed cells are actually clear. So this is after the 3rd wash of the red blood cells, so you can see the supernatant is clear, so that means that they're thoroughly washed. So I'm going to decant both samples here. And then come back over to the lab. And then what I'm going to do is I'm going to have a new tube here, so I want to make sure everything's lined up. So I've got Brutus and Brutus here. So I'm going to take 0.02 mils of the packed red blood cells and put it in my new tube here, and then I'll add the 0.98 of saline. Okay, let me just finish the math and then I will get back. Okay, thanks. Okay, so I'm going to add my 0.2. And then I'm adding my saline. So I'm going to set that one down, do the same thing for Carmel. the saline here. And then what I'm going to do next is take a few drops of this mixture and then a few drops of the serum. And for the major cross-match, I'm going to do serum from the recipient, and then for the minor, serum from the donor. So what I'm going to do now is take a dropper. I'm just checking to make sure I'm getting the right amounts of everything. So 2 drops of the recipient serum and 2 drops of the donor cells. So I'm going to use one of my conical tubes here. So this is the minor cross-match. This is for Carmel. So what I'm going to do is take a few drops of her serum. And then a few drops of the mixture from Brutus. And I'm going to set that on my rack here and let that sit. I'm going to do the same thing here. So this is the major cross-match, so I'm going to take serum from Brutus. He's the recipient. And then the packed cells from Carmel. Gently shake that, and then I'm going to do the same thing with controls, so doing Carmel's serum and her packed cells together, and same thing for Brutus. And then I'm going to let all 4 tubes sit here for about a half hour, incubate at room temperature, and then we'll come back. So all 4 of the tubes sat at room temperature for about 30 minutes, and then I centrifuged them. So both of the controls, you can see there's really no obvious hemolysis, which means that there wasn't any contamination. And then the same thing for both the major and the minor cross-match. You really don't see any hemolysis there either. Shake them gently to see there's really no obvious signs of autoagglutination either, so I'll examine them under the microscope. I already placed a drop of each sample onto a slide and put a cover slip on, so I'll just head right over to our microscope here and take a look. I'm going to look at the major cross-match first. So far, I'm not seeing anything obvious. Just kind of scan through the whole, through a few fields. I'm not seeing any autoagglutination on this slide, so I'm going to switch to the same thing and look at the minor cross-match. Same thing here, not really seeing anything. So based on the fact that there was no hemolysis or autoagglutination, we would say that Carmel and Brutus would be a match for blood donors. So now we're going to do the skin scrape on Brutus. So what I'm going to do is I'm going to shave a small patch back here just so we can get down to the skin. He's being very brave. Good boy. Then what I'm going to do is I'm going to take mineral oil and I'm going to put some drops on the slide here. I'm going to open up my scalpel blade. I'm going to hold it gently here between my thumb and my forefinger, and I'm just going to get some of the oil on it. And then I'm going to hold this at a perpendicular angle to his skin and just kind of scrape it until I get a little bit of capillary blood. Sorry, Brutus, you're being a good boy. And this is to make sure we get a deep scrape. If it was a superficial scrape, I would either just use a tape prep or I would just scrape some of the hair and the skin right off. Good boy, buddy. Good boy. He's like, what are you doing? Okay, I'm starting to see a little bit oozing there, so I'm gonna stop. And then I'm gonna transfer the material here onto the slide where the mineral oil is. And then we'll come back and look at that under the microscope. So this is the slide from Brutus's skin scrape. We're gonna look at this next. This one we don't actually have to stain, we can just go straight to the microscope. So I'm gonna place it on the stage there, and then I'm gonna look at it under low power to start. Things like Sarcoptes you can see pretty easily on low power. So far, no signs of mites, maybe just a little yeast, which is kind of what we expect for him being a dog with chronic allergies. I look through several fields, kind of scan the whole slide, make sure I'm not missing anything. So next what I'm going to do is I'm going to put a cover slip on it, and then I'll read it, same thing again, this time only under, whoops, the high dry. This would be to check for Demodex. You can't see them as obviously. Don't see any mites, again, just a little bit of yeast, some hair follicles. So good news is Brutus' skin scrape is negative. He doesn't have sarcoptic or demodectic mange, so that's good news for his owner. So that's the end of my video project for this lab class. It's been a lot of fun to shoot. Thanks for watching.